Hello, in this video, we're going to close our discussion about lipids and we'll talk about cholesterol. And it fits into a completely different category, okay, in it's a major class of lipids, but it's not a ketoacyl lipid. It's in the isoprene category. And this is the fundamental backbone of sterols, and we'll see how cholesterol looks like. And it's just, there's the sterol ring, and then it's got all these extra places where there's a double bond. So if you look how it looks, if you see how it looks, got the extra double bond, and then some side chains of the sterol ring. And if you care to know about the origin of the name, people knew. I mean, they identified from bile, which is choli, and then it's solid, something in the bile. It's an alcohol because of this hydroxyl group, right? Hence the name cholesterol. Cholesterol has fascinated chemists and biologists for decades. The chemical synthesis, all in a test tube, was done. It was at the time. It was a heroic effort by Bob Woodward in the 60s. Got the Nobel Prize for it. So it was a major milestone. The total synthesis of cholesterol in the lab. What does it do? It does a lot of things. Cholesterol is actually essential. We can't live without cholesterol. In terms of our membranes, these are the phospholipids, the polar groups facing the outside are hydrophilic, and you have the side chains, the two fatty acids in the inside. If that's your standard membrane, let's say. So this is a hydrophobic environment. This is where cholesterol goes and wedges between the hydrophobic side chains of the phospholipids, and in doing so, it actually regulates the overall structure of membranes. Okay, so it regulates. So it has very important essential functions, structural functions in membranes, the permeability, how fluid the membrane is. So these are very important. These are not the only functions, and also extremely important functions are the biosynthetic roles. Okay, cholesterol is actually a building block for even more complex molecules, all the steroid hormones in mammals, bile acids, which we will see, and other stuff. Right. In addition, cholesterol itself can be used to decorate, let's say, proteins. They get modified the same way we saw proteins getting phosphorylated or other things added to them. You can add cholesterol, okay? So, or sugars, for example, we've seen that too. So cholesterol is one of these decorating entities that can be added to other things such as proteins. So you can't live without it, but if you have too much of it, as we all know, that's problematic. All right, so let's look at the synthesis. We're not going to look at it in detail. It's too complex. It's got 39 steps actually in the cell. So we're just going to see a schematic and point out a specific point where PUTSIS has a huge biomedical significance. That's the statin drugs, how they work to block cholesterol synthesis in the cell. So here the building block is isoprene, which is made from acetyl-CoA. Okay? And you have, first you start with from acetyl-CoA, you make HMG-CoA, which we have seen already this reaction in, when we talked about ketogenesis, the ketone bodies. Once you make HMG-CoA, which looks like this, then you reduce it with HMG-CoA reductase, and it is that enzymatic step very early on in cholesterol biosynthesis that is blocked by statins. Right? If you don't have statins and you allow things to go on, eventually you end up with cholesterol, and this you see here many arrows. You know, as I said, it's almost 40 steps, but it bifurcates at some points, and you can see interesting things here. For example, ubiquinone. Okay, how that is made in the cell, and we've seen ubiquinone. It's so important in electron transport chain, right? So that can serve. That's a bifurcation there that can lead to ubiquinone synthesis. So it's not, so this is the pathway is called overall the mevalonate pathway. And because the product of HMG-CoA reductase, which is inhibited by statins, as I said, they're very potent inhibitors of HMG-CoA reductase, is mevalonate. mevalonic acid. So the whole pathway is called mevalonic acid. Other compounds are known to inhibit the pathway, the phosphonates. And as I said, the pathway is extremely important for a variety of downstream products, not only cholesterol and ubiquinol, which we already mentioned. You get heme too. Now, in terms of statins, the inhibitors of HMG-CoA reductase, it has been estimated that just statins alone in the general population, which has been used, I don't know, since the 80s, has added maybe as much as 10 years to the lifespan of the US population, let's say. So extremely important. And even though they do have some side effects, they're actually fairly safe drugs, very safe drugs. Now, in addition, as you saw here, you get, actually you don't see it here, but from cholesterol, you can make bile acids, cholic acid. Here, so actually from cholesterol, you can make cholic acid shown here. And from that, then, you can make all kinds of stuff, all kinds of bile acids in the bile that are extremely important for digestion. Okay, the way you digest fat, for example, you have to coat the fat that you take from the food with these bile acids. And so from cholesterol, you get cholic acid and chenodeoxycholic acid, and then you keep modifying them further. And depending on what you conjugate them with, you get a whole variety of them. So all of them, all the conjugations happen in the liver, and all of them are extremely important and necessary for digestion. If you have certain diseases, let's say pancreatic cancer, or your gallbladder doesn't work anymore. So you have to take some of that stuff as drugs, exogenously. Right? In addition, so the cholesterol then, how does it get transported in the circulation? You can find them in the chylomicrons, which we have already seen in the digestion of the dietary fat. These are the triacid glycerols, the T sign, so they are inside the chylomicron. So you have some phospholipids. You have on the outside, you have some proteins that make the outside, the casing, let's say, of the chylomicron, and within it, actually, you can find cholesterol too. Right? So if you ask what's the source of cholesterol in our cells, you can make some, okay? And the endogenous biosynthetic pathway that we just discussed, that's the one that's blocked by statins. And that helps a lot. However, we do take a lot of cholesterol from the diet, and it's found in these chylomicrons originally, then eventually it makes its way into LDL, okay? And how does LDL come in the cell? There is a specific LDL receptor, okay, that brings it in, and then that's how you get even more cholesterol, right? This was how cholesterol from LDL gets into the cells was a major target of research. People were trying to find it, and I'm just showing you this slide because Michael Brown and Joe Goldstein at UT Southwestern were the ones that discovered it, and they got the Nobel Prize in 1985. So it was a huge deal to understand. So fellow Texans, it was a huge deal to understand how cholesterol comes in from the circulation. So that's all I had to say about cholesterol and lipids, and we'll close at this point with this video.